Welcome to Study Loop.
Okay, so problem 28 is all about the mitogen-activated protein kinase cascade, right? And so these are triggered by our RTK receptors. So those are the ones with an intra and like extracellular component. Okay, and so what's going to happen is the MAPK pathway basically it sort of has this RAS, RAF. So this is like this whole cascade, right? Going through these proteins here. Then we have the MEK protein and the ERK protein. And this cascade, this phosphorylation cascade, this is also sort of called this this is a parenthesis, this MAPK pathway. This is what will trigger cell division, right? And cell division equals growth. So this is gonna cause growth or division, however you want to think about it. Okay, and so we have a list of what would cause uncontrolled growth, right? And so we want to see what sort of mutations would cause more of this to happen. And so just looking through all these options, I think sometimes it's easier to pick out the things that are incorrect, right? So for example, one of the items listed under A is a loss of function in the MAPK pathway, right? So if we have a loss of function of our MAPK, okay, well, so that would be this not functioning, okay? But if that doesn't function, that causes growth. So there's no way that mutation would lead to uncontrolled growth, right? So then we know that A can't be one of our answers, right? And so if we look at say B here, one of the ones listed in B is to have the I kappa, the Greek kappa here, be permanently bound to the NF kappa B. Now, if you remember, the I kappa B is an inhibitor of the NF kappa B, and this guy will lead to sort of starts your transcription of your DNA, right? So this is going to transcribe. And so if this is permanently inhibited, it can never enter the nucleus and start that process. And you certainly need more DNA to duplicate, right? So this, the I kappa B permanently bound would certainly not allow for this additional and extra growth. Okay, and then if we want to take a look at our choices in D, okay, so this one has a loss of function mutation as well in both the RAS and RAF, right? But we need those. We need those to get to this growth and division that's part of our MAPK pathway. Okay, so this RAS / RAF protein loss would not lead to more growth. Okay, so that sort of just leaves us with C, right? But if our RAS protein was unable to hydrolyze, then it's not going to sort of do what it needs in the cascade. And if we have a loss of function, of the inhibitor, right, then our NF kappa B will always be able to enter the nucleus. So we would sort of get that function back. So that checks out. That's good. And if we gain a mutation in the genes for the pathway, so there's gain of function somewhere in here. So that means it's more functional and creating more growth and division. That checks out. That would certainly help. And then lastly, an unregulated phosphorylation cascade, right? So that's this cascade is sort of happening in here. And if that's unregulated, that means it has the potential to sort of go out of control and not check itself. So all the options here in C do seem to check out, and that is our answer, is letter C.